Hi everyone, my part is the effect of the estrogen E6AP complex on the transcriptional regulation of the NPY1R gene to study the pathogenesis of Angelman syndrome. So to understand what that all means, it's first important to know what Angelman syndrome is. So Angelman syndrome is a sex-linked recessive disorder that primarily affects the nervous system. So what we typically see in Angelman syndrome patients is that we see severe memory loss, loss of intellectual disability, and even severe speech impairment. And though Angelman syndrome is a relatively rare disorder, it affects a large number of people with about 200,000 cases per year. So the pathogenesis of Angelman syndrome is really simple. You have a mutation on this gene called UBE3A. And the UBE3A gene is really important to make this protein called E6AP. And the E6AP protein regulates about 22 different genes, which are important for normal brain activity function. So what happens is that when you have a mutation in the UBE3A gene, you prevent the production of the E6AP protein. Therefore, you lose regulation of all these 22 different genes, and that's what causes Angelman syndrome. So we know that when you lose the E6AP protein, the effect is Angelman syndrome. But what we don't know is the mechanism as to how the E6AP protein regulates the different genes it regulates. So previous studies have tried to figure this out. And previous studies have found that estrogen binds to the E6AP protein to bind to certain estrogen receptor sites. But no one has ever been able to link that certain function to a certain gene with Angelman syndrome. So that's what my project is going to be. In order to link that E6AP estrogen complex to a certain gene that E6AP regulates. So in order to do this project, I took the 22 different genes that E6AP regulates, I took the DNA nucleotide sequence and inserted it in common databases such as Jasper and ClusterBuster. And I specifically wanted to scan for the promoter site of each of these genes to find an estrogen receptor site on these genes. So after scanning all these genes, I found that the NPY1R gene, which is a gene that E6AP regulates, I found the NPY1R gene has an estrogen receptor site in this promoter. And what the NPY1R gene does is that it produces neuropeptide receptors for normal memory function. So when you lose regulation of the NPY1R, you lose your memory function. So I specifically did 3 different phases of the experiment of the project to test the hypothesis that the E6AP protein binds to estrogen to go to the receptor site of the NPY1R gene to allow normal transcription of the NPY1R gene to occur. So the first phase of the experiment was the luciferase phase. And that point was to determine how effective is estrogen binding to the estrogen receptor site of the NPY1R gene. So what I did was that I took the DNA sequence of the estrogen receptor site and the NPY1R gene, and I inserted it in a luciferase plasmid. And then I treated the cell with luciferase and compared it to a negative control and a positive control. So after comparing it to a negative control, we found that when you use estrogen, there was almost a 12-fold increase in luciferase activity, showing that when you treat the NPY1R gene with estrogen, the estrogen receptor has a very high binding efficiency to the estrogen receptor site. So then I wanted to look more on a molecular level to see how estrogen plays a role in the transcriptional level of the NPY1R gene. So in order to do this, I took the NPY1R plasmid with the estrogen receptor site and I inserted it into neural 2A cells, which are hippocampal cells that are typically found in mice. And after inserting the neural 2A, the NPY1R plasmid into the neural 2A cells, I treated the cell with either estrogen or no estrogen, and then I compared it to a negative and positive control. And when compared to a negative control, when the NPY1R plasmid was treated with estrogen, we found almost a 4 times increase in mRNA levels compared to the negative control. So that shows that estrogen somehow plays a role in allowing the normal NPY1R transcriptional level to occur. So then I wanted to see what would happen if we take the E6AP protein out of the equation. So that was the 3rd phase of the experiment. And what we did was that we specifically got a RNA which was called SIE6AP. And what the SIE6AP does is that it kills the E6AP protein. So it takes the E6AP protein out of the equation. So we treated neural 2A cells with estrogen or no estrogen and the SIE6AP. And then we compared it to another set of cells that were treated with typical normal controls and estrogen and no estrogen. So we found that when you take estrogen when you take the E6AP out of the equation, you will have almost a 36% decrease in mRNA levels. So that shows that if you take E6AP out of the equation and have no estrogen, there's a 36% decrease in mRNA levels. So that proves the hypothesis that the E6AP protein binds to estrogen to go to the receptor site to allow for normal transcriptional level to occur. But if we lose the E6AP protein, and even if we lose the estrogen hormone, then we see that you lose the transcriptional activity of the NPY1R gene, and that leads to a 36% decrease in mRNA levels. So that shows and proves my hypothesis that the E6AP protein binds to estrogen to go to the estrogen receptor site of the NPY1R gene and allow for normal transcription to occur. So now we found a specific mechanism as to how E6AP protein regulates the NPY1R gene. So the application of the experiment is that now that we find a specific mechanism of how E6AP regulates NPY1R gene, we can make certain treatment models for Angelman syndrome which are not typically found in the market. And right now there is no cure for Angelman syndrome. So this project allows future research and future clinical research to find a cure for Angelman syndrome. For example, we can find another protein that's similar to the E6AP structure, or we can find another E6AP estrogen complex that allows for that normal NPY1R transcription to occur. So instead of E6AP and estrogen, we can have a totally different set of estrogen hormone complex or protein hormone complex to allow for a normal transcriptional level. And for more future research, and it's research that I'm doing right now, is to determine how the E6AP protein regulates the other different genes that E6AP protein regulates. Thank you for listening to my project. Have a good day.